Welcome to today's histology lecture on the topic of elastic cartilage. Elastic cartilage is a specialized type of connective tissue found in various parts of the body, particularly in areas that require flexibility and support. It is characterized by the presence of elastic fibers, which give it unique properties not found in other types of cartilage. In this lecture, we will explore the structure, development, functions, and pathological changes associated with elastic cartilage, as well as techniques used for its identification. So, let's dive into the fascinating world of elastic cartilage. Elastic cartilage is a type of supportive connective tissue that provides elasticity and shape to certain anatomical structures. It is predominantly composed of chondrocytes, which are the primary cells responsible for maintaining and synthesizing the extracellular matrix of the cartilage. The matrix consists of collagen fibers, proteoglycans, and elastic fibers. The abundant presence of elastic fibers differentiates elastic cartilage from other cartilaginous tissues. Elastic cartilage is found in several locations within the body, such as the external ear, pinna, auditory tube, eustachian tube, epiglottis, and the larynx. Its unique composition allows it to withstand repeated bending and deformation without undergoing permanent damage. Elastic fibers in elastic cartilage are primarily composed of the protein elastin, which confers elasticity and resilience to the tissue. Synthesis and assembly of elastic fibers involve a multi-step process. Initially, fibroblasts within the cartilage produce a precursor molecule, tropoelastin. Tropoelastin undergoes enzymatic cross-linking, facilitated by enzymes like lysyl oxidases, to form organized elastic fibers. The process of cross-linking imparts specific mechanical properties to the elastic cartilage, such as stretchability and recoilability. The development of elastic cartilage begins during embryogenesis, with the differentiation of mesenchymal cells into chondroblasts. Chondroblasts secrete an extracellular matrix rich in proteoglycans and collagen fibers, which act as a scaffold for cartilage formation. As development progresses, chondroblasts mature into chondrocytes, which maintain the cartilage tissue's integrity throughout life. During development, certain genes and signaling pathways play crucial roles in determining the fate of mesenchymal cells towards elastic cartilage differentiation. Transcription factors like SOX9 and SOX10, as well as growth factors like TGF-beta, are involved in regulating elastic cartilage development. Elastic cartilage exhibits regional variations in structure and composition, depending on its location within the body. For example, in the external ear, elastic cartilage is densely packed and extensively branched, providing shape and supporting the framework of the ear. In contrast, in the epiglottis, elastic cartilage is more loosely arranged, allowing for flexibility during swallowing. Each variation in structure and composition is specifically adapted to the functional requirements of the anatomical site in which the elastic cartilage is located. The regenerative capacity of elastic cartilage is limited compared to other tissues in the body. However, researchers have been exploring various strategies to promote cartilage regeneration and repair. These include tissue engineering techniques, such as the use of biomaterial scaffolds and stem cells, as well as the manipulation of growth factors and gene expression to stimulate chondrogenesis. Although still in the experimental stage, these approaches hold promise for future therapeutic applications. Certain genetic disorders can disrupt the normal structure and function of elastic cartilage. One well-known example is Marfan syndrome, which is caused by mutations in the fibrillin-1 gene. Individuals with Marfan syndrome exhibit abnormal elastic fibers, leading to skeletal abnormalities, cardiovascular complications, and ocular defects. Other genetic disorders affecting the synthesis or assembly of elastic fibers can result in similar manifestations. Understanding the underlying genetic mechanisms behind these disorders is crucial for diagnosis, management, and potential therapeutic interventions. As we age, elastic cartilage undergoes various structural and functional changes. These changes include a decrease in elastin content, deposition of additional collagen fibers, and a gradual loss of tissue elasticity. These age-related alterations can lead to reduced flexibility, increased stiffness, and a decline in the overall functional capacity of the affected anatomical structures. Elastic cartilage plays a crucial role in sound transmission within the human body. In the larynx, elastic cartilage forms the vocal folds, vocal cords, which are involved in voice production. The elastic nature of these vocal folds allows them to vibrate and produce different pitches and tones. Disorders affecting elastic cartilage in the larynx, such as vocal nodules or polyps, can significantly impact voice quality and clarity. Elastic cartilage plays an essential role in the respiratory system. In the trachea, it helps maintain the patency of the airway, preventing collapse during breathing. Similarly, in the bronchial tree, elastic cartilage provides structural support and helps regulate airflow. Dysfunction or damage to elastic cartilage in these regions can lead to respiratory complications and breathing difficulties. While elastic cartilage is primarily found in the head and neck regions, it also has specific roles within skeletal structures. For instance, the intervertebral discs, which separate the individual vertebrae in the spine, contain elastic cartilage. This cartilage allows for flexibility and shock absorption, enabling smooth movements of the spine. Pathological changes in elastic cartilage can arise due to various factors, including trauma, inflammation, and genetic disorders. These changes may include degeneration, calcification, fibrosis, or the formation of abnormal lesions. Pathological alterations can impair the normal function of elastic cartilage and surrounding tissues, leading to a range of clinical symptoms and complications. Immunohistochemical staining techniques are commonly employed to identify and distinguish elastic cartilage from other tissues. These techniques rely on the binding of specific antibodies to the components of elastic fibers present in the cartilage. By utilizing various antibodies against proteins such as elastin or fibrillin, histologists can visualize and characterize the elastic fibers in tissue samples. Immunohistochemical staining techniques provide valuable diagnostic information in clinical settings, aiding in the accurate identification and assessment of elastic cartilage abnormalities. In conclusion, elastic cartilage is a unique tissue with remarkable properties including elasticity, flexibility, and support. Its biosynthesis, development, functions, and pathological changes are important to understand for accurate diagnoses and effective treatments. Techniques like immunohistochemical staining aid in detecting elastic cartilage. Thank you for joining me today.